Deep within the Atlantic Ocean, approximately 4,000 meters down, lived a sad little anglerfish. Her name was Glowy McChompface. Glowy was dark purple in color with a large head. Her mouth was enormous and crescent-shaped, with sharp and translucent teeth. As most anglerfish are often less than a foot tall, Glowy was heavily judged for being a large 3.3 feet in length, especially by her peers and also by other Midnight Zone inhabitants.
Despite being pretty much alone, Glowy always longed for something more. She had one dream: to see the world as the fishes above did. She wanted to witness the golden sun melting into the horizon, to feel the warmth against her skin, and to escape, only for a minute, the endless blackness of the Midnight Zone that had always been her home. Glowy, like all other female anglerfish, has a distinctive piece of dorsal spine that extends over her mouth, which kind of looks like a fishing pole. On the top of her dorsal spine, or on her esca, lay the only light she ever had, a dim, shallow light that allowed her to distinguish between food and rocks.
To understand Glowy's light, we need to explore the science behind bioluminescence, a phenomenon driven by genetic mutations. A genetic mutation is a change in the DNA within an organism. More specifically, it can be a change in the sequence of the nucleotide bases in DNA, called a point or gene mutation, or it can be a mistake within a whole segment of DNA on a chromosome, known as a chromosomal mutation. It is important to note that a point mutation can stop the production of a protein in an organism, which could be very bad because proteins are necessary in vital functions for all living organisms.
Chloe's light-producing lure in her esca is not caused by a genetic mutation from her own body, but rather from a genetic mutation of the symbiotic bacteria that live near her lure. These bacteria lack the genes needed for independent survival, such genes that synthesize proteins and sense nutrients. Because these mutated bacteria produce light in her esca, Chloe then provides nutrients and a safe home for them in return. The LUX gene, that is found in a cluster of genes known as the LUX operon, is affected by the bioluminescence mutation, specifically the LUX-A and LUX-B genes, which encode subunits of the luciferase enzyme. The luciferase enzyme has the task of emitting light in the bioluminescent reaction. Due to the uniqueness of the bioluminescence mutation, the creation of a protein is not stopped in the bacteria that are Chloe's lure, but rather the bacteria produce special light-emitting proteins like luciferase. All this science may be confusing, but just know that this is an explanation of what provides Chloe with a small light at the tip of her esca, one of her only friends in the Midnight Zone other than Tammy.
This is Tammy, the super rare Homo sapien. Luciferase is an enzyme. It interacts with a substrate which is oxidized luciferin to therefore affect the rate of the chemical reaction. As a result, a byproduct is created called oxyluciferin. But most importantly, the reaction creates the light at the tip of Glowy's esca, which she loved above all things. This is where Glowy's dream will come to life. As we know, her sole wish was to see the giant light that looked exactly like hers, but much bigger. The sun. Glowy was done letting time pass while her dream became more and more distant. So, she decided to go for it. Glowy was going to try and reach the sun. So she took a big gulp, said goodbye to Tammy, and headed upwards.
While Chloe's making her way up through the Midnight Zone and towards the Twilight Zone, let's discuss how the bacteria in her lure provide her with her special light. In order to create the protein luciferase, the bacteria need to go through protein synthesis. The first part of protein synthesis is transcription, which takes place in the bacteria's single circular chromosome located in their cytoplasm. Firstly, helicase begins the process of unzipping the DNA double helix on the gene that codes for luciferase. Then, RNA polymerase comes in and helps RNA nucleotides line up along the template strands of the DNA, following the complementary base pairing rules. Complementary base pairing is when each nitrogenous base pairs with its complementary nitrogenous base. For example, the nitrogenous base guanine always pairs with the nitrogenous base cytosine, and adenine always pairs with thymine. After all the nucleotides have been assembled, a single strand of M or messenger RNA is created. Next, as the mRNA peels away from the gene then rewinds back to its original double helix shape. The purpose of this mRNA being created through transcription is because it acts as a copy of the genetic material, so that the DNA of the bacteria doesn't get damaged when it goes out into the cytoplasm for the next step of protein synthesis. If that was really confusing, think of transcription like Glowy deciding to chase the sun. Her dream is copied from her thoughts into action, just like DNA is copied into mRNA. This is similar to how transcription works, as DNA is being transcribed so the original copy or her original dream is not affected by a failed execution.
It's been a while since we've checked in on Glowy, so let's do that now. Here she is, a solid 2,000 meters up from where she started, looking stronger and more determined than ever. Now, getting back to protein synthesis, Glowy's bacteria have now successfully created a mRNA strand. This strand will travel as a messenger, carrying its genetic information, which has instructions to make the protein luciferase to the ribosome.
At the ribosome, translation will begin. The ribosome is made of R, or ribosomal RNA, and proteins. Its subunits will hold mRNA and tRNA together during translation. Once at the ribosome, T or transfer RNA anticodons will match up to complementary mRNA codons and therefore attach the specific amino acid that they are carrying to a peptide chain. A codon is a sequence of 3 nucleotides on an mRNA strand that codes for a specific amino acid or signals the end of protein synthesis. On the other hand, anticodons are placed at the bottom of tRNA molecules. Additionally, they play a huge role in translation by acting as the key that recognizes and binds to specific codons on the mRNA. In order to ensure the correct amino acid is added to the continuously growing polypeptide chain, and as you know, amino acids are the monomers of proteins.
In the case of many other amino acid chains, and in luciferase also, the first mRNA codon codes for the amino acid methionine. Eventually, one codon, called a stop codon, will be reached on the mRNA strand that will signal the process of protein synthesis to a stop. And then the amino acid chain will compress and morph into luciferase's specific folded shape. I know translation may seem really complicated, but try to think of it like Glowy's actual journey up to the sun, where her determination, or mRNA, is read and turned into movement, just as ribosomes read mRNA to build a protein. Way through the twilight zone, Glowy realizes that while she's a bit different from her peers in the midnight zone, she's vastly different from the creatures in the twilight zone. She's shocked that none of them have their own lights. Glowy wonders why she's different from them, and that would be why any living thing is different from one another. Genetic mutations. We already touched on the mutated bacteria in Glowy's lore that produce her light, but let's dive deeper into what specific type of mutation these bacteria may possess. As we've established already, bioluminescence doesn't alter the function of a protein, but rather it creates a new one, called luciferase. However, point mutations can still occur in the genes that code for luciferase. Of the point mutations that can be found in Glowy's bacteria which affect bioluminescence, a popular one occurs in the LuxA gene. This gene can have a missense substitution mutation, which could change the amino acid histidine to arginine, therefore making the light produced by Glowy either much weaker or stronger.
Glowy has now made it through the twilight zone and is in the sunlight zone currently, meaning she is only a short 200 meters from the sun. She can see the rays brighter than ever. In fact, it was so bright that she can't even make out her own light anymore. Despite being severely tired, Glowy pushes through and makes it to the top of the ocean. Saying that she was amazed is an understatement. Glowy was awestruck. How bright and shiny the sun was, she thought. It was everything that she could have ever hoped for. But suddenly, Glowy felt a horrible pain in her body. We'll save the specifics on the rest of this situation, but just know that the pressure change really got to her. While Glowy may not be here with us anymore, she at least passed happily, knowing that she had accomplished her one dream, witnessing the golden sun melting into the horizon. Just as Glowy's dream is completed, when she reaches the surface, a completed protein takes shape at the end of translation, fulfilling its purpose.